Good afternoon. Welcome to all of you here in Missouri and those who are watching on the video. One of my pleasures as NIH director is the opportunity to come on Wednesday afternoons and be able to introduce the speaker. But there's no greater pleasure than when the person who's speaking is somebody you know well, and in this case, somebody that I had a chance to be a partial mentor for back a few years ago, like 15. And it's always a joy to see how somebody that you've played some small role in helping to train makes a wonderful success of their own career path, and then you get to bring them back and embarrass them a little bit with an introduction. So that's what I'm here to do this afternoon for Dr. John Carpten. John got his PhD at Ohio State, and he came to the fairly fresh out of the blocks intramural program of the National Human Genome Research Institute back in 1994. When Jeff Trent had just been recruited as the scientific director, somebody that I brought with me from the University of Michigan to establish an intramural program in genomics. It was probably taking a little bit of a chance to show up at that point and expect that things would actually go well, but they went extremely well. And no small part in the cancer genomics arena because of John. John had prodigious energy and does today, perhaps even more so, and had a hunger for trying to get answers about hereditary and somatic contributions to cancer that has driven his career forward over that time, now over some 17 years, to where he is today as the Deputy Director of Basic Research and Senior Investigator at the Translational Genomics Research Institute in Phoenix, otherwise known as TGen. Over the course of that time, he has accomplished a number of very significant observations and publications in the area of cancer genetics, beginning with when he was still here as a postdoc, co-leading an effort that led to the first identification of prostate cancer susceptibility using linkage analysis on families. I had the great pleasure of working with John in an effort to focus this effort especially on African American families, which had otherwise not really had much attention paid to them. And through that came about the African American Hereditary Prostate Cancer Consortium, which John had a major role in helping to get started and which continues to this day as a very interesting and important enterprise to try to understand why it is that prostate cancer happens at a higher frequency and tends to be more severe in individuals of African American background. In his work at TGen, he has continued to make strides forward in understanding factors involved in cancer, including work that you might have seen in terms of identification of a HOX gene that seems to carry in a New England Journal article a substantial increased risk of prostate cancer if a certain point mutation is present, as well as very interesting findings about AKT and its role in lung cancer. So it's always a joy for me when I pick up publications or see what's going on in the world to see that John continues to make this kind of wonderful contribution. I think he's going to give you a really interesting presentation about some of the things that are now going on at TGen with comprehensive molecular profiling, trying to connect what's possible now in the research arena with what's going on in the clinic without having the barriers that sometimes get in the way, prevent that from becoming possible in real time for the benefit of patients. John is both a wonderful scientist, a wonderful human being. It's really a joy to see him back here today to give this presentation. So please join me in giving a warm welcome to my friend, John Carpten.
Thanks, Francis. Well, thanks, Francis, and to say that that was a humbling introduction is an understatement, and I've always appreciated your leadership and your guidance and your mentoring through the years, along with those other individuals who have played a significant role in my career development. So with that said, I do have quite a few slides, and I want to make sure I can get through as many of these as possible. And again, thanks to those who were able to make it here and to those who are viewing remotely. Today I'd like to talk to you about some of the work going on at TGen, a pipeline or platform that we've developed to help, as Francis said, connect what we can do as it relates to molecular profiling in cancer and comprehensive profiling, and to link that intimately with what's going on in the clinic, particularly around advanced cancer, to really try to form a true nexus between the bench and the bedside. And so I want to bring to bear some of the technologies that we're utilizing and the approaches that we've taken or developed, and walk through a few vignettes on some of the, whether people want to call them anecdotal observations and results, or some of the significant observations that we've seen in some of the patients, and then finish up with some of the areas that we think are critically important in trying to build out these pipelines and these platforms so that they can be more widely applicable. So the basic premise is that many cancer therapeutics are currently aimed at specific targets that are in many instances associated with genomic alterations, and this has been somewhat of a paradigm shift in pharmaceutical development, where in many cases drugs were identified more serendipitously, where a drug has some effect in a cell line during a screen, and then that agent would be further developed and moved through the pipeline. Currently, a lot of drugs are being, and compounds are being developed specifically to target certain molecular perturbations that have occurred within a cell. One of the poster children, of course, being CML and the BCR-ABL fusion in response to Brian Drucker and Charles Sawyer's work in the development of imatinib and dasatinib. Also in breast cancer, HER2/neu, we all know, in response to drugs such as trastuzumab and lapatinib. In red, I've highlighted a couple of areas in which my laboratory has had the honor of working in collaboration with scientists at the Mayo Clinic in Scottsdale, identifying mutations that converge on the NF-kappa B pathway, and showing that these alterations are associated with response to the proteasome inhibitor bortezomib. And Dan Von Hoff's work, along with his colleagues at the Mayo Clinic in studying basal cell carcinoma, identifying activation of sonic hedgehog pathway in many cases through mutations in the patch gene, and the use of vesmodegib, the smooth-end inhibitor. Other interesting observations, of course, are seen around triple-negative breast cancer or basal breast cancers, and particularly those harboring germ-line mutations in BRCA1 and BRCA2. I also think this expands to somatic alterations in these particular genes, in response to a class of drugs called PARP inhibitors, many of which are currently being developed in the clinic, and a new class called PARP trappers. There are a number of examples showing some first-in-class drugs in phase 1 studies that have showed outstanding efficacy in the clinic. The drug crizotinib, which was a MET inhibitor, which works in lung cancer patients that have non-small cell lung cancer patients harboring the EML4 ALK translocation. And this is just sort of tumor burden on the y-axis. And here are a series of patients in yellow. These are the ones harboring the EML4 ALK translocations, and vemurafenib in metastatic melanoma patients harboring BRAF V600E mutations, and vesmodegib in patients with basal cell carcinoma with hedgehog activation. The context is generally not as straightforward as looking at one particular alteration, as we've seen with EGFR inhibitors such as erlotinib and gefitinib. Patients that have these alterations or treated with these EGFR inhibitors tend to do well. However, when these mutations are present or wild type in the context of patients that have mutations in KRAS, these patients actually do worse. So understanding the genomic context of tumors can help in potentially determining the best course of treatment for patients. So the question we've asked at TGen early on was, is molecular profiling a rational approach to increase the options available to oncologists for treating these patients, particularly those patients who have failed standard of care, who have advanced chemo-resistant cancer. So one of the first studies to show the benefits of molecular profiling in the treatment of cancer is a study that we at TGen call the Bisgrove trial, because it was primarily funded by one of our good friends and philanthropist, Jerry Bisgrove, where Dan Von Hoff and Mike Bittner and Bob Penny performed a study looking at about 106 tumors, performed gene expression profiling and some limited immunohistochemistry, and treated patients based on molecular alterations, and then measured progression-free survival after first-line therapy, and was able to show that they could actually see a significant increase in progression-free survival when they treated patients based on the molecular profiles. This study, although landmark, was faced with several levels of criticism, one being the limited technologies that were used, another one being that they focused on a number of tumor types instead of focusing on one, and then finally that it wasn't a randomized study. So again, thinking about the molecular measurements and different types of mechanisms of alterations, we see that many of and this is just a small list of known cancer genes, but what we know is that many of these genes can be altered through various mechanisms. HER2 can be amplified, it can be overexpressed at the protein level. EGFR can be amplified, mutated, overexpressed. KRAS as well. We can look at tumor suppressors like P53, P10 that can be deleted or mutated. Tumor suppressors like RB and P16 can also be hypermethylated at the promoter. And then we also know that some genes are activated through structural events such as translocations and fusion transcripts such as BCR-ABL, TMPRSS, and EML4-ALK. Previously, we've had to rely on a series of different technologies, methods, and core capabilities that were required to be present to assess these different types of alterations. If we wanted to look at point mutations, in many cases we'd use a sequencing or genotyping technology to identify copy number changes like amplifications and deletions. We'd use CGH microarray technologies. For RNA expression, probably everyone in the room has run an Affy array before to look for gene expression differences. And to identify structural events like EML4-ALK, we still use FISH and microscopy. But what we've seen is the development of amazing technologies over the last 5 to 7 years, particularly next generation sequencing technologies. And these technologies are amazing because, of course, they've increased our ability to get to a human genome by order of magnitude in terms of time and price. But for cancer, they're amazing because they allow us to make all of the necessary measurements to identify any of the types of alterations that one might look for in a cancer. Point mutations, we can identify across the genome. Copy number changes, we can assess amplifications and deletions. Gross rearrangements like translocations, breakpoints, and inversions can be identified using these technologies. And if we have RNA available, we can also perform very detailed transcriptional profiling, measuring expression at the base level, the exon level, identify particular isoforms, and also look for fusion events. So these provide the platform for very comprehensive genomic interrogation in a single assay. So utilizing these technologies, we've now built out a platform that is currently under FDA-based analytical validation, where this pipeline typically starts with the patient and the doc and consent through biopsy, sample and analyte extraction and QC, deep molecular profiling using next-generation sequencing analysis, and utilizing a proprietary analysis tool that will take a somatic compendium report. That's a report that will contain all of the somatic events within a given tumor and match the events against a drug-gene relationship database to generate a report that can then be discussed at a molecular tumor board, and then we can provide those recommendations back to the treating physician. We currently have this pipeline, again, going at around 21 days, and we're pretty sure that we can get it down to 14 days with improvements in our analytical pipeline. So the question now becomes, can we use deep profiling using next-generation sequencing as an approach to increase the options available to oncologists? And so instead of performing standard whole genome sequencing, we actually use a different approach, which is a genome-wide approach. And we typically want to sequence both the tumor and the normal, particularly at the DNA level. And we do whole genome, but it's a long insert whole genome methodology. Here, instead of sequencing fragments that are roughly 300 bases, we sequence fragments that are about 1,000 bases, which increases our ability to identify structural events. So although we have low base coverage, we have very high clonal coverage across breakpoints. So by sequencing only to 50x coverage, we have, 5x coverage, we have 50x physical coverage across the genome, which is very powerful for determining copy number changes, identifying breakpoints, translocations, and inversions. We couple that with, at the DNA level, with deep exome sequencing. I have roughly 100x coverage here. Technologies now allow us to get to about 250 to 300x using our current approach. And this gives us great sensitivity to identify point mutations down to about 10% allele frequency. We believe we can go down to 5%. And again, having RNA available, we perform RNA sequencing of the tumor and a reference. There's no such thing as a perfect reference in differential expression. So we use a different series of methods for gene expression profiling to identify overexpressed or underexpressed genes, but also as a powerful way to improve our ability to detect mutations, particularly in tumors that have low tumor normal ratios. Identifying a mutation at low frequency in the tumor DNA, sometimes we'll see that mutation in the RNA, and it lends credence to the likely credibility of that particular mutation. We're leveraging the Illumina HiSeq 2500 system. We were one of the first 4 labs in the world to have one of these set up and running, and they're now commercially available, which will get us to normal tumor whole genome analysis within a 27-hour time period. We've built out a very comprehensive somatic bioinformatics pipeline that goes from standard data processing and management through typical secondary analysis, alignment, and variant detection, all the way through identifying the genomic and clinically relevant events down to a report. Part of the reason we've been able to improve this pipeline, and now our current research pipeline is about 12 hours, is because of a collaboration we have with Dell Computing, where we've been able to get access to some of their more innovative Sandy Bridge processing technologies. Again, we've built out this platform. We initially called it JEDI for genomics-enabled drug inference. Some people love it, some people hate it. Jeff actually went to high school with Steven Spielberg, and so we're always worried about running into problems using the word JEDI. But here we have a somatic compendium report from a particular patient's tumor, which will have all the mutations, the translocations, the differentially expressed genes. We've also developed an in-house proprietary drug-gene relationship database, which contains evidence and information that relates different genes and different perturbations to known therapeutics. And these two databases are merged through a heuristic algorithm that we call Turbine to generate these clinical JEDI reports. And these will contain the gene of interest, the type of biomarker, the specific aberration, the evidence, whether it's direct or inferred, the particular drug or class of agents that might be relevant to that alteration, and the indication whether that drug should be given that context, a no, yes or a no. These tables have evolved over time, and now we even provide information such as the frequency of that particular mutation and the sequencing pileup, and a series of other ancillary pieces of information that provide more information as the tumor board meets to make appropriate recommendations. So next I'd like to walk through a series of vignettes where we've applied this type of approach to real patient samples and live fire exercises and looking at some of the outcomes of these studies. The first study is a project we did where we were focusing on patients with metastatic triple-negative breast cancer. It was an amazing study. The clinical lead was Joyce O'Shaughnessy, who's at Baylor Sammons in Dallas, Texas. She's also part of US Oncology and Texas Oncology and Life Technologies, where we're able to apply this genomic knowledge towards treating patients with metastatic triple-negative breast cancer. So we did, in this particular study, use a whole genome approach and whole transcriptome approach. We were able to, in this study, which was pretty much our first endeavor, to develop an integrative analysis pipeline as well as a genomics medicine knowledge engine that we've been able to modify and improve through time, and to assess the outcomes in the context of the genomic and transcriptional profiles in these patients. And I'd be remiss not to mention David Craig, who sort of my partner in crime in developing this pipeline, who's our Deputy Director for Information Sciences at TGen. The initial study was 14 patients, again, with clinical diagnosis of metastatic triple-negative breast cancer. I think it was really amazing that, unlike some studies, we had a very nice diversity among our patient population, where among the 14, 8 were classified as white, and 6 were classified and self-identified as Black and or African American. You look at the ages, we had a few, really young women in the study. We were able to assess tumor cellularity, so we had very, very high quality specimens that also led to really, really nice analyte. Here, all the RNA samples had RINs of 8 or above. Like the TCGA, we're able to really understand the mutational landscape of triple-negative breast cancer, maybe not with the numbers, but it was interesting, even with 14 patients, we had very similar numbers. The most frequently mutated gene in triple-negative is P53, again, which was shown by the Cancer Genome Atlas breast cancer group. RB mutations, P10s, the alpha-catenins, which I do want to talk about a little bit. I'll provide a few slides because there were some interesting observations around alpha-catenin, and ERB4 alterations. Again, we had RNA sequencing data, so we were able to look at differential expression, and we were able to compare the RNA-seq results from our patients versus a set of normal controls that were ethnicity and age matched. And one of the things that fell out among the vast majority of patients was we always tended to see this G2M checkpoint cassette where FOXN1 was always one of the most highly overexpressed genes. Other genes that fall within that cassette are aurora kinases A, B, PLK, TTK, which is MSP1, and we're actually working on developing some new compounds around targeting TTK for specifically in triple-negative breast cancer. Talented postdoc in my lab, Nicole Lavender, is heading up those studies. We also were able to look at, compare the gene expression profiles against the Layman program, which is the group Kim Pyatt-Paul at Vanderbilt developed an algorithm to determine triple-negative breast cancer subtypes, and the vast majority of our patients fell into the basal-like classification, but we also had a series of patients that were deemed immunomodulatory or the Chuck-Perot Claudin low group, and we also had at least one patient that had the luminal AR subtype. One of the other interesting observations was around P53 mutations, and this was something that didn't fall out in the TCGA data, was that we tended to see significant allele-specific expression of the mutated allele at the RNA level. So when you're just looking at the DNA level, many of the mutations fell out to look as though they were heterozygous, where you could sort of see about a 50-50 ratio of mutant to normal. But what we would almost always see at the RNA level was very, very high expression of the mutated allele. And this was interesting because none of these patients had LOH at the 17q region encompassing the P53 locus. What was also interesting was that we were seeing this type of expression of the mutant allele even in the context of normal contaminating stroma. So you'd think there'd be some normal P53 expression in the stroma, but we were seeing these real high levels of P53 expression of the mutant allele even in the context of contaminating stroma. And we were able to search through and find a couple of papers that had showed that there was this interesting phenomenon where mutant P53 may actually be turning off the expression of wild-type P53 in the surrounding stromal tissue. It's a phenomenon that hasn't been deeply studied, but other people have seen this, and there was a paper about a year or two ago looking in glioblastoma in cancer research that saw somewhat of a similar observation. I think it's an interesting observation that warrants further analysis. Another interesting observation were these alpha-catenin homozygous deletions, and this is just the read pile-ups of one of the patients. Here's the alpha-catenin locus and the flanking gene SIL1. You can see in this patient's tumor this huge deletion where there are very few sequencing reads across the encompassing the alpha-catenin locus. And here we can see a second patient. I think what's interesting about this is that Elaine Martis's group back in 2010 had sequenced a single African-American triple-negative patient. And this patient, they had the primary, the met, and a xenograft of the metastatic tumor. And they also identified a homozygous deletion in that patient that encompassed the alpha-Katina locus. And it's really interesting in that there's 3 of 16 triple negatives with homozygous deletion of alpha-Katina, but both of our patients were African-American. So that would be 3 out of 7 triple negative patients that are African-American that have homozygous deletions across the alpha-Katina locus. We actually were trying to filter through the Cancer Genome Atlas data. They had about 100 triple negatives in that study, but very few were African-American. So we really would like to really look into the real frequency of alpha-Katina perturbations in African-American triple-negative breast cancers. We know alpha-Katina is involved in cadherin and cell invasion and metastasis. So is there a context around perturbations or loss of alpha-Katina related to the more invasive phenotype that we see among African-American women with triple-negative breast cancer? So again, another study that we're following up on. So these are Circos plots. Many of you have seen these before. Pardon the resolution, which will show the genes that are perturbed around the outside, the copy number changes, and the magenta lines represent translocations and structural events. And these are just 6 different Circos from 6 of the different patients. And I wanted to focus on patient 2 here. And you can see this is a 60-year-old Caucasian patient with recurrent disease after 7 months on gem-carbo-aniparib. So this patient actually had primary disease that came back. She then went on standard of care for metastatic triple negative, which is gemcitabine plus carboplatin, and she had also been randomized in an aniparib PARP inhibitor study. And we sequenced the tumor. And you can clearly see a series of copy number changes, some amplifications here. She had a series of deletions, but also a series of translocations and breakpoints. And if you look closely, you can see this just sort of explosion of events out of chromosome 7. And so further analysis of this patient's sequencing data showed that this actually was a series of events occurring across 3 different chromosomes that formed what's called a double minute. So you had breaks here, 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 and here that formed one small circle. And these extra chromosomal circles are autonomously replicating. So you had this little circular DNA containing different bits and pieces of different chromosomes that contain the BRAF locus. So this patient had a tumor just spitting out tons of BRAF through the replication of this double minute. We were able to get a back clone that encompassed the BRAF locus and perform FISH, and you could see the validation and the confirmation of these double minutes in her cancer. This patient, we went on to assess clinical utility of the genomics data, the BRAF amplification in double minutes. There was also high-level gain across the HRAS locus and INPP4 downregulation. INPP4 had been studied by Lou Cantley's group and shown to be a tumor suppressor in breast cancer that acts through the P53 kinase AKT pathway. And so the molecular mechanism here we believe driving this cancer was RAS-RAF-MEK-ERK, but also with concomitant activation of the AKT pathway. And so the thought was it would be great if we could identify a combination trial where this patient could be treated with an inhibitor that would inhibit both RAS-RAF-MEK-ERK as well as P53 kinase AKT-mTOR. It was important to clearly validate these events before we went forward. And in this case, we were able to validate INPP4 downregulation and BRAF upregulation. We were able to work with the group at START, Tony Tolcher, the group in San Antonio, and the group at MD Anderson and others who had a clinical trial with GSK where they actually were looking at combination therapies to hit P53 kinase AKT-mTOR, RAS-RAF-MEK-ERK, and expanded, I believe it was an expanded phase 1. There weren't a lot of patients benefiting from that study. We talked to them about the genomic analysis that we had for this patient, worked with GSK, were able to get this patient on that combination therapy, and lo and behold, she had what Joyce called an otherworldly response, greater than 85% decrease in tumor burden with only 2 cycles on combination trametinib, which is actually FDA approved now, and at that time, the GSK 214 AKT inhibitor. And one of the common themes we saw in this particular study were a convergence of mutations within these two pathways. And although this wasn't something necessarily new, we actually were able to act on it therapeutically and show benefit for some of the patients, where we saw a number of events affecting RAS, MEK-ERK, and we saw a series of events involving PI3 kinase AKT-mTOR, again in the same patients. And we had RAS amplification and overexpression, a patient with an NF1 homozygous deletion that also had a P10 homozygous deletion. We had the patient with the BRAF and INPP4 downregulation. We had a patient with FBXW7 homozygous deletion and activation and amplification of FGFR1. So we saw this sort of exciting dual pathway activation, and we're able to act on it. So the basic outcomes of this study were activation of these two pathways in about 40, if not 50% of patients. Some of the events I just mentioned, we were among the first to show possible efficacy of the MEK-AKT combination, and there are currently several trials ongoing currently looking at combination MEK plus AKT inhibitors in triple-negative breast cancer. Joyce O'Shaughnessy is running one, and working with Pat LaRusso at Karmanos Cancer Institute to hopefully get another one off the ground. And the results of this study were published in Molecular Cancer Research earlier this year and highlighted as a significant study. The next application of this, the application of this technology in a case study is around metastatic cholangiocarcinoma or bile duct cancer. These cancers can be associated with liver flukes in some cases, and in other cases, they're sporadic and non-liver fluke. And this is work we did with Matesh Borad, Alan Bryce, and Rafael Fonseca and Keith Stewart and our partners at the Mayo Clinic. So this particular patient, 50-year-old male with supraclavicular lymph node Mets primary bile duct cholangiocarcinoma. The patient had been previously treated with gemcitabine plus cisplatin. We sequenced this tumor, and here's the Circos plot, and you can see a series of mutations, a series of translocation events, and intrachromosomal rearrangements. One of the interesting things was that the path reports showed about 25 to 75% tumor cellularity across a series of biopsies. But the sample that we sequenced, actually looking at the allelic fractions in the sequencing pileup, suggested that we only were dealing with about 10%, if not 15 to maybe 10% tumor content. We identified 35 somatic coding mutations. 2 genes were in cosmic. None were in any of the known panels, commercial panels that are available. I won't call names. 1 was flagged for therapeutic context. I'm going to focus on that. 1 copy number event, nothing flagged. RNA-seq didn't flag anything. Structural events didn't flag anything. And you can see the CGH plot, it's pretty stable genome, nothing sort of jumping up or down. But we identified this 1 mutation, which is a nonsense mutation in a gene called ERRFI1. Pardon the resolution here. I probably should have zoomed in on the mutation. But this is the patient's normal genome. This is the normal exome, tumor exome, and RNA sequencing. And interestingly, we only saw the mutation in the exome at about 11% frequency, but 80% in the RNA. Again, another example of an important tumor suppressor like P53, where we see preferential expression of the mutated allele. Again, no deletion across the locus, but preferential expression of the mutated allele. This is a very, very important gene. Again, it's not in any of the cancer panels, but why, I don't know. Because ERFI1 is actually an endogenous inhibitor of EGFR and ERB signaling. Biochemical and cell-based analyses confirm that the interaction contributes to EGFR interaction. So ERFI1 sits in the dimerization pocket of ERB receptors and prevents them from dimerizing, thus preventing them from becoming active. And this study actually went on to, by Zhang et al., went on to show that if you put in wild-type ERFI1 or MYC6, you can significantly decrease EGFR phosphorylation. However, a series of mutant constructs were not able to abrogate reduction in phosphorylation. Our good friends at the Van Andel Institute, and we didn't know this at the time, actually showed that MIG6 was also a tumor suppressor. They actually developed a transgenic model of MIG6 by knocking out MIG6, and lo and behold, the knockout mice developed adenocarcinoma in a series of organs, including the bile duct, right? Which was just another very compelling evidence about the importance of, relating the importance of MIG6 to bile duct cancer. We stained for total EGFR, and you can see there's really nothing here. However, when we stained for phospho-EGFR, we see lots of staining, importantly membrane staining, which was indicative of EGFR activation. Again, just a model of where it sits. It sits in this dimerization pocket and prevents homo and heterodimerization of ERB receptors. So the therapeutic context here was ERFI1 loss as a modifier through an inactivating mutation and preferential expression of the mutated allele, and potential sensitivity to EGFR inhibitors. And so we highlighted and we also know, and I showed earlier on, that KRAS mutations in the context of EGFR activation can prevent the efficacy of EGFR inhibitors. And having the exome data, we were able to show that there were no activating mutations or oncogenic perturbations within RAS-RAF-MEK or PI3 kinase AKT-mTOR. So an EGFR inhibitor was recommended after CLIA validation of the event. And this is tumor before, and this is tumor after. So about 40 or 50% tumor reduction, and this is the metabolic activity of the tumor essentially went completely away after a few cycles on erlotinib, and the patient has a real nice stable response to EGFR inhibitor. So we sequenced 6 metastatic cholangios. We identified the ERFI1 EGFR context, but the other incredible observation were fusion events and translocations, including the FGFR2 oncogene. And I won't run through this other than to show this little line of red sequencing reads that are indicative of a translocation event encompassing the FGFR2 gene, and in this case, its partner, BIC1. Now, Rule Shanayan's group at Michigan also found these as well in cholangiocarcinomas, and was able to do some really elegant functional analysis to show that these fusion events are actually oncogenic. We validated these by Sanger. We have 3 different partners, all fusing to the last intron of FGFR2. So essentially, you get all of the kinase domain of FGFR2 binding to these different genes and fusing to these different genes. And Arul was able to go on to show that, interestingly, the regions that are retained within these genes are oligomerization domains. So what you're getting is lots of FGFR2 heterodimerizing together. We were able to show significant overexpression of FGFR. And there was another paper that showed that deleting the carboxy terminus of FGFR2 is actually oncogenic. So we were lopping off the carboxy end of FGFR2, fusing it to oligomerization domains, activating FGFR2. So in these particular patients, FGFR2 activation, sensitivity to FGFR inhibitors was the therapeutic context highlighted. And at the time, there were a series of agents being developed that target, in many cases, FGFR and VEGF receptor. However, some are far more specific to the FGFR receptors, some of those being pazopanib and ponatinib. At the time, ponatinib was being touted as probably the best FGFR inhibitor, but it was sort of in the last phases of being approved by the FDA, and they wouldn't allow us access to the drug because they didn't want to threaten their FDA approval. So the first patient actually went on pazopanib, and here's the tumor, one of the tumors before pazopanib, and this is 4 months later, and this patient had a relatively stable and a significant response over about 7 or 8 months. Later on, that patient actually was put on ponatinib, and you can see this is looking at tumor density, densitometric measurements. This is tumor before, lots of cancer, and you can see the tumor is almost completely melted away after about 6 weeks on ponatinib. And here's more tumor, same patient, tumor. So this has led us to think about perhaps a bucket-style trial for specifically around cholangiocarcinoma, where we take a series of patients and actually place them in a bucket based on molecular profiling. So patients with NRAS might go on a MEK inhibitor, patients with FGFR fusions might go on FGFR inhibitors, and patients with EGFR activating events like ERFI1 inactivation might go on an EGFR inhibitor such as erlotinib or some of the others. Another way to look at this would be to do a randomized study where you place patients, or an adaptive study where you place patients in the appropriate bucket and your control arm is standard of care chemo. So we're trying to figure out what the best design might be for a study, but we have a number of clinicians who gastrointestinal clinicians and oncologists really excited about these discoveries, wanting to do clinical trials. So again, I hope what I've been able to show is the power of using a more comprehensive approach, use the long inserts to identify translocations, RNA to identify fusions, and to provide confidence for mutation calls, particularly in the context of low tumor cellularity, and deep exome sequencing for identifying mutations that might be in a lower content within a specimen. And just to further show the importance of RNA, it can also detect critically important therapeutically relevant isoforms. So this is a patient with pediatric GBM, and we were able to show that is actually a leaf ramini. The patient harbors a germ-line P53 variant. That's an ACMG variant of unknown significance, but predicted to be deleterious, and we now know that the mutation is transmitted in the family. But interestingly, the copy number analysis from the next-gen sequencing data also showed a significant high-level amplification of EGFR, which does occur in GBM. But it was interesting that we were able to provide this information to the oncologist, who then went on to ask us, okay, it's EGFR amplification, but is it the V3 variant of EGFR, which is really a highly oncogenic active variant of EGFR. But if all we had were exome data, we might have been able to identify the amplification if we had CGH or whole genome. But if we didn't have the RNA, we would not be able to know whether or not the V3 variant was being expressed, because this particular mutation is known to occur in pediatric high-grade gliomas and is relevant with response to targeted therapies in this particular tumor type. And we were able to show that RNA sequencing data, these are sequencing reads. Here you can see reads mapping to exon 1 of EGFR and spliced over to exon 2, 3, 4, 5, 6, 7, and 8. And so these are your standard EGFR full-length transcripts. However, you can see a series of reads that map from exon 1 and skip all the way over to exon 8. And this is the indicative profile for the EGFR V3 variant. So by having the RNA data, we were able to show with relatively high sensitivity and specificity that not only did the patient have a high level amplification of EGFR, but was expressing the V3 mutation as well. So through our comprehensive approach, we've identified these events, including in triple-negative breast cancer, and looking at some of these other studies, the FGFR rearrangements and fusions, ERFI1 mutations, and sensitivity to EGFR inhibitors. Again, this very, very therapeutically relevant gene is not available in any of the known cancer panels, and we're hoping it eventually makes its way because we know that the group in Spain actually found a ERFI1 inactivating mutation in a patient with GBM. RNA data also provides us additional power for identifying mutations and looking at allele-specific mutation, again, the isoform. So we really believe in the comprehensive approach is most suitable for capturing the compendium of somatic events that might occur within any given tumor to reduce the potential for false negatives when applying this technology in the clinic. And lastly, I just want to talk, because one of the major issues we know in cancer is heterogeneity. We know that there was a really impressive paper that came out in the New England Journal about a year and a half ago showing the complexity of any given tumor specimen. And I think it goes beyond a tumor. I think there's also heterogeneity within the context of the patient's cancer. There's a tumor and then there's the patient's cancer. And so we've been studying heterogeneity from a series of different approaches. And one I wanted to talk about was, which we describe as clonal tides. and we were able to show this through whole genome sequencing in patients with multiple myeloma, where we actually had a single patient, and we followed the patient longitudinally from diagnosis all the way through plasma cell leukemia, which ended up killing that particular cancer patient, unfortunately. And so the patient was initially treated with Revlimid plus dexamethasone, and you could see a significant reduction in tumor burden and then a relapse here. The patient then went on Velcade, a proteasome inhibitor. You could see a nice response and then another relapse. And the patient went on another Velcade plus response, and then melphalan plus a series of other genes, other therapies, response, and then plasma cell leukemia. The patient succumbed to their disease. And again, it was an amazing study because we were able to capture the primary tumor, pre-treatment, the tumor at first relapse, the tumor at second relapse, at third relapse, as well as the plasma cell leukemia, and sequence all four of those tumor specimens. And what we were able to show was that there was, and I'll just sort of walk through some of this, and I just get to this, that there were two clones at diagnosis. One made up about 95% of the tumor, the other made up about 5% of the tumor. And so what happened was the primary tumor was very responsive to dexamethasone plus Revlimid, but the minor clone was resistant. And so what happened was there was an initial response and a first relapse, and this is the tumor that came back. And this tumor was treated with proteasome inhibitors and responded. And then there was a relapse. Guess what happened? The first tumor came back, right? Now, if the doc had known that there was this clonal tide, then perhaps the patient could have went back on Dex plus Rev, and maybe there would have been a response. But it's very, very rare that a doctor will go back to the same treatment twice, because we tend to think of resistance as a linear sort of evolution of tumors that have become resistant to the first line of therapy. And so we're able to show this clonal tide that you went from diagnosis to first relapse, back to a tumor that was almost the same, and then back to another tumor that was similar to the first relapse, which was just an amazing study that just goes to show that heterogeneity is not as simple as taking a tumor, breaking it into pieces, and saying that the pieces are different, but the patient's full context of cancer should be taken into account. So in summary, we're building out and refining this pipeline so that we can apply these technologies to provide options. Again, we're not making treatment decisions. We're providing options that the doc can use to make a recommendation. We're working on a current randomized clinical trial through our Stand Up to Cancer study, which is specifically looking at melanoma patients with metastatic disease who are non-BRAF V600E carriers. We're working with the FDA very intimately, and we're submitting an umbrella IND as well as an IDE type of submission to be able to bring to bear the genomic technology on these patients to select the therapy that we believe is most relevant given the patient's genomic context or molecular context. Some of these include locking down our rule selection for matching genes to drug. And again, these documents actually are going to be submitted later on this week, and we're hoping for the best. So we're refining our pipeline, and we're applying it to a series of studies. Again, I talked about the metastatic triple-negative study, the cholangiocarcinoma study, the SU2C study, which will be a randomized phase 2 clinical trial. We have another study in advanced glioblastoma that will also be a randomized phase 2 clinical trial. And then we have a longitudinal study to look at the natural history and heterogeneity, specifically in multiple myeloma. So this one I'm really excited about. It's being led by a really fantastic junior investigator, Jonathan Keats. Will be 1,000 patients at diagnosis, followed through. We're collecting tumor pre-treatment, and then we're going to collect tumor at relapse. And we're performing deep genomic profiling, whole genome, exome, RNA, on the primary as well as the relapse tumor, so that we can look at the natural history of this disease during the course of different treatments and clinical trials. Of course, there are a number of considerations and aspects that require further discussion and refinement. Again, sample QC, tumor cellularity, and heterogeneity. We have to understand that we're only treating the tumor sample that we biopsied. Trying to make any other assumptions is moot. Can we deal with needle biopsies versus full resections? We're currently able to perform our deep molecular analysis using 18-gauge frozen needle biopsies. Study designs, strengths and weaknesses of using panels. They're usually fast, they're very specific and sensitive because you can sequence deep, but there's a specific false negative rate because you're gonna miss all of the things that you didn't assess. The strengths and weaknesses of a comprehensive approach: low coverage, long insert genomes. The deep exomes provide higher sensitivity, RNA-seq direct targets, and supporting integrated information. But one of the weaknesses is that it's a little more lengthy and a little more costly. Clinical trial design, we've seen adaptive trials like the BATTLE trial in lung cancer. We actually want to work more towards randomized phase 2 studies to determine whether or not molecular profiling is as good as, worse, or better than the current standard of care in the advanced setting. One of the challenges is always access to pharmaceutical drug pipelines and portfolios. We're lucky to work with some amazing clinical collaborators. One of the other issues is combinations. We're seeing multiple events occurring in patients, and we need to combine some of these treatments, which you just can't do on the fly because you got to have at least minimally dosing studies, PK/PD, to know whether or not you can combine two agents. The other important thing that we're doing is we're generating tumor grafts where we can. So we're taking the tumor out of patients and planting them in animals, and we can actually test the efficacy of these combinations, at least in a preclinical setting, to determine whether or not they're going to be effective or not. Technologies, we've seen incredible improvements that provide benefits for sensitivity and specificity. We need standards specifically for looking at somatics. We're working with Justin Zook and Mark Solid at NIST, who've developed this program called Genome in a Bottle, where they've developed NA12878 Ceph DNA as a reference for CLIA for germ-line, but we need a reference for cancer. And we're working with some of our collaborators at one of the technology companies, as well as Mark and Justin, to develop a cancer standard for CLIA sequencing. And again, it's important to get all of this into CLIA, which we're currently doing. And speed. Some people, I remember Francis, the Genome Project was 15 years to get to one genome, right? To get this done in the clinic, the oncologists need to get this data within 2 to 3 weeks, and that's what we keep hearing. And we believe we're there, but I'll be honest, there was a big push towards a $1,000 genome. I could care less at this point. Give me a 24-hour genome. If it costs $10,000 to get to 24 hours, I'd rather have that than a $1,000 genome that takes a month to get to. So at the end of the day, the promise is to deliver the most appropriate drug to each patient's tumor based on a combination of molecular and evidence-based medicine. The challenge, of course, is will this approach lead to improved outcomes and effectiveness without sacrificing efficiencies to the healthcare system? So I'll stop there and entertain any questions. Thanks.
Thanks, John. We can have the lights up, and if people have questions, please approach the microphones in the aisles, because that way the people who are watching on the web can hear as well. Roland.
Have you been able to see any therapy-induced escape mutations?
Oh, absolutely, absolutely. And again, the question is, do those mutations arise, or is it an evolution? Meaning, did the bulk tumor, or was there one clone that acquired a new mutation based on the therapy? Or was that mutation already there in a minor clone that you just couldn't detect because it only represented 1% to 2% of the tumor mass? Or you didn't sample that particular part of the tumor, or maybe you didn't sample that tumor at all, right? You take a biopsy from the liver, the patient also has a lung met, and all you biopsy is the liver. Does that mean that you've actually also represented the lung met as well? So I think the challenge is to really understand, are we looking at tumor-acquired mutations, or are we looking at clonal evolution, meaning there was a clone that had already acquired that mutation? Now, there's been some amazing work by a number of investigators, whether it's Dan Haber, who's looked at circulating tumor cells, looking at lung, and showed the EGFR T290M, I think, that it was there in the circulating tumor cells. But when you look at the tumor mass, it was there at very, very low percentages. And Matt Meyerson's work, almost 10 years ago now, where he took a pleural effusion from a patient with lung cancer and showed that the T290M, T790M, I can't remember, was there, but it was only in about 2% of the tumor. So initial assessment of the tumor, of the patient's bulk tumor, didn't show the mutation. When we looked at the pleural effusion and sequenced like 15,000x coverage using, I think at the time it was 454 or something, right? They saw the mutation. So it was there in a minor clone. So again, the question is, are there mutations that are arising specifically from the tumor being bathed in chemicals? Or are those mutations already there and would cause the mutation to occur in the first place? Great question, though.
John, with respect to the 3 out of 7 African American women with triple-negative breast cancer and alpha-Katina, homozygous alpha-Katina deletions, do you know whether there was a germ-line mutation associated with that, or was that entirely spontaneous mutations? And if the latter, how do you account for that?
They are somatic. They were somatic events, meaning the germ-line was essentially wild type across the region, and the tumor deleted both copies.
So why would that happen preferentially in African American women? What's the concept here?
The concept, or my hypothesis, is that there could be a haplotype that is associated with breakage at that region. And in a particular context, there's hyper breakage at that region. And if you happen to be the unlucky individual that is homozygous for that haplotype, you get breaks of both copies. And we have to understand is that the deletions are different, right? There's 2 deletions that sort of overlap, and then you get the region of homozygous deletion. So there are 2 different alleles, right? 2 chromosomes. In some cases, the whole chromosome, one copy of one chromosome has been completely deleted, and then you have the deletion that encompasses the alpha-Katina locus. So my feeling is that we see this type of ethnic enrichment, I think, in other diseases, PML and Latinos, and the breakpoints that occur in the retinoic acid that form the retinoic acid translocations and fusions in PML. They're very specific breakpoints that you see almost exclusively in Mexican Americans. So the question is there a germ-line haplotype that is related to genomic instability at that region and preferential breaks occurring there? And so to tease that out, I don't know how we get there. We need to look at the frequency of the breaks, and then we have to take a careful assessment of the germ-line and compare that to the germ-line of patients who don't have the breakpoints.
Yeah, I guess it's also possible if you don't see anything cis to the mutation that it could be a transmutation of some kind.
Right, and the question is that if there is a haplotype, is it in cis or is it in trans, right? And these are the types of things that we need to, it's tough to get specimens. TCGA took them all. Yeah.
Hi. I'm wondering when you're giving recommendations for treatments for combination therapy, whether you can get to the point where you're suggesting relative amounts of one particular drug versus another, instead of just a yes or no, give that drug or not. And I also have one more question after that.
Yeah, no, that's a great question. And, dosing, drug sequencing, do you give one therapy first and pulse and then give the second therapy? And if so, in what order? I think, there was a study out of Yale that showed this amazing study, and I think it was triple-negative breast cancer, looking at EGFR inhibitors plus doxycycline and showing that if you hit with dox first and then hit with EGFR, you see this massive response. But if you give them together or in the other order, you don't see any response. And so that is an active area of research, and I guess the simple answer to your question is no, we have not done that yet. Doing combinations at all is a challenge in and of itself, and I think, but we're going to have to battle and tackle combinations because the vast majority of these tumors have concomitant activating mechanism and events, and we're going to have to target multiple things to see, the best responses.
And my final question is, when you have a mutated gene that seems to be, from your RNA data, overexpressed versus the non-mutated gene, could it be that the RNA that's mutated is less susceptible to degradation? Or do you have any clue what could be the...
there are multiple mechanisms, right? The wild type allele could be methylated. There could be uniparental disomy, meaning you've lost the normal chromosome and duplicated the mutated chromosome, right? So it looks like there's no real LOH. right? But uniparental disomy does occur. Or it could be an issue of RNA stability, absolutely.
Hi, so I work on sickle cell disease in Africa, and one of the problems that we've found is that when we've tried to replicate some of the reported loci that have been found in African American populations, we haven't been able to find them with the same number of frequencies or prevalence in our population, and it's been very difficult to validate the results of new loci that we've identified in Africa, in African American populations. So can you comment on your experience, whether you've done any studies in African populations with breast cancer? And secondly, what do you think would be the strategy for fast-tracking that kind of research? And what would be the chances of making sure that African populations get access to the drugs that are available in this country?
Wow, that's a mouthful. It's a lot to address. So I'm not surprised. I don't think, we've been studying, the genetics of populations of recent African ancestry for years. I've, had some amazing collaborations with Rick Kittles, who's currently at University of Illinois, Chicago, who's one of the pioneers of ancestry, and particularly at recent populations of recent West African descent. And so I'm not surprised. I think there's an immense amount of heterogeneity just within Africa itself, much less between Africans, West Africans, and African Americans. And I think we have to look at populations. I think it'd be great if we could first determine ancestry by looking at ancestry informative markers rather than saying, here's a class of African Americans, here's a group of West Africans, and asking the question, can we validate? Is there a way for us to look at the African Americans and ask, is there a cohort or group among them that have ancestral alleles or patterns that look more West African? And then focus on that group and ask whether or not we can validate in that group or not. But I'm not surprised that we don't see, and we've been lucky, and one of the studies I'm really excited about is we have a Komen Promise grant with Max Wisha at the University of Michigan and Pat LaRusso, looking specifically at breast cancer stem cells, triple-negative breast cancer. And within that study is one of the aims is to look specifically at women from Ghana, and that part of the work's being led by Lisa Newman and one of her clinical fellows who are collecting specimens, triple-negative breast cancer specimens from women in Ghana. We're implanting those tumors in mice. So we'll have tumor graft models, and we're performing deep sequencing. Those samples are actually on the sequencer right now. And hopefully Evelyn's going to present some of it at the Disparities meeting in Atlanta. But you're right, because one of the things we want to do is we've got a small number of African-American triple-negative sequence now. So the question is, how similar or different will the tumors from Ghana look compared to African-American women and the tumors that we've looked at here? And then finally, I think what you're talking about is policy. Making these approaches and these technologies applicable for everyone. God, I'm at NIH, but I don't work for the government anymore, so I can say it. Perhaps the Affordable Care Act will help us get at some of that. I'm hoping it does. Will it be the only thing to help us get there? No. I think we're talking about culture. We're talking about the way we do practice medicine today. I think all of these are factors that we're going to have to deal with. It's great to see John Ruffin's group. I had a chance to talk to John today, amazing conversation, hoping that he will, part of his vision is to help us understand and how to get there, understanding disparities, understanding the challenges we have in dealing with disparities, and trying to make these technologies available to everyone. Our first study with triple-negative breast cancer was almost 50% African American, and if nothing else we had done from that study, I felt that in itself was a win. That we actually are able to work with clinicians who understand the impact of the disease in all people and try to provide access to these cutting-edge technologies regardless of race or socioeconomic status.
So there's a reception in the new FAES space up the hall here, which you're encouraged to come and continue the conversation with John Carpten over coffee and cookies. But I think right now we should thank our speaker again for a very stimulating presentation.